after yesterday. It's quite a day. Thank you.
No nudity.
Thanks to everybody. No repeats, please. It was enough.
No nudity today.
Yeah.
No frontal nudity.
No frontal. Well, I don't know what his plans are.
Please, no. I won't sign on the defense.
So we have an exam, and all of you probably know the rules, but let me just quickly review how we do it. Generally, he gives a talk. If you have questions, try to save it to the end so he has some momentum. If there's something clearly that you see that's not clear or crazy or wrong or something, don't hesitate to put your hand up. At the end, if you save your questions, we'll have it open to the floor, and then we'll ask everybody to leave, and we'll have our colleagues on the committee stay here and ask him if he knows anything. So thank you very much. And Gaurav is going to give the talk. You can read as much as I can. It's about proteins at interfaces.
Thank you, Mr. Bhatt.
You're on.
So thanks all for turning up for my defense. I've been involved with the study of protein adsorption at solid liquid interfaces and going from the few of the projects. So I'll just start with the introduction to protein adsorption, followed by different studies I have been involved with at RTI in Professor Bhatt's group. So to start with, we have used a simple osmolyte molecule to self-assemble on the surface and make it make the surface hydrophobic surface protein resistant and works for small proteins. Next, I'll discuss. Most of the time in adsorption studies, people use self-assembled monolayers or polymeric substrates, but real metals are sometimes ignored. So I've chosen transition metals along a period, and I'll discuss the adsorption of 3' blood proteins on metals and how effective is the chemical method to remove it. So alkaline SDS have been shown to be perfect for removal of proteins. I'll show you the results of this study. Then protein adsorption studies are quite numerous over the years by many scientists, but stability of covalently attached proteins or the monitoring the folding unfolding of covalently attached proteins is the studies are lacking. So we developed an assay using AFM atomic force microscope in force mode to develop a method to monitor folding and unfolding of these proteins. In the interest of time, I won't be able to discuss the rest of the projects. So this was in collaboration with Professor Kumar's group in Colombia, and this was the adsorption and characterization of surface adsorbed proteins, followed by some work which I did for Professor Doddick's group, that was an ATR FTL measurements. So just to start with, proteins are biomolecules which are marginally stable, and that's how they are made by nature. Perhaps that's the way they function. So the folded unfolded transition, we can write the folding energy, which is quite low. So they are marginally stable, and so they can be easily unfolded by different kind of perturbations in like temperature, pressure, or organics, chaotropes, and surfaces. So my study is how surfaces perturb the protein structure, and why is it so important? I'll just show it to you. So protein adsorption is just like it's similar to polymeric adsorption, where there's a fast step of diffusion from bulk solution to the surface. So at this point, the protein can dissolve back, or it can undergo conformational changes. So this is the fast step, and this is the slow step. And it has been shown by previous members of our group and others that these conformational rearrangements are primarily related to the structural changes in protein. So there is a predominant changes in alpha helical content to beta sheet content. That's what has been shown in the past. So why is the study important? It's important both in medical and non-medical applications. In medical applications, there are numerous kinds of surgical instrument, catheters, implants, where you don't want your proteins to be sticking onto the surfaces and causing complement activation or other problems, inflammation problems. But in non-medical unit operations in bioprocess industry, protein adsorption can be desirable or can be undesirable. So if you want to, in chromatographic columns, suppose you need a surface which has specific affinity for some proteins, so it is desirable. But at times it is not desirable, like in filtration where it fouls the membrane. So removal of protein from the surfaces depends on protein stability on surfaces, and that's why it is quite relevant. So just to start the first study, so we used a osmolyte molecule, the very well-known osmolyte molecule, trimethylammonium oxide, with self-assemblies on the surface, and I'll show how can we use it to resist adsorption of small proteins. So there has been many approaches to make surfaces protein resistant in the past. We have used inorganic polymers, PEG, and there was studies by Professor Belfort in the past using betaine, and Professor Kane, sugar-based and surfactants. And these are the common thumb rules which have been pointed out by Ostini and Whiteside's group, that typically the surfaces which are hydrophilic in nature, which are typically neutral, and which can actually have the hydrogen bond acceptors, that means the water is stable on the surfaces, they tend to shield proteins from the surface. So these are the common thumb rules. All these approaches, most of them are covalent modifications of the surface. That means the surface is not regenerable, or you have to spend a high amount of energy to remove that chemistry from the surface. But we have used the self-assembly or the non-covalent attachment, so the surface is degenerable. So that's the novelty or the difference in this work. So quickly, what are osmolytes? Osmolytes are have been identified as small organic molecules which are often present in high concentrations in organisms which live under stress. So deep in the sea, where there is a lot of hydrostatic pressure, these organisms like shrimps and fishes, they have high concentration of these molecules, and they tend to protect protein structure. How do they do that? There is like many hand-waving arguments, but no exact mechanism is known. But by various groups, it has been identified that the transfer energy of these molecules to the protein backbone is unfavorable, so they tend to keep themselves excluded from the protein and keep the protein well hydrated. That's how they function. So among the class of these osmolyte molecules, there is like amino acids, sugars, and carbohydrates like 3 halos. But TMAO is known to be the best osmolyte, or it has the maximum capacity of protecting the structure. So looking at the TMAO molecule, it is a very small 75 Daltons small molecule, and at that length scale, it has the beauty that it has a hydrophilic end and a hydrophobic end. So no such molecules. We surveyed other molecules too. At this length scale, it's really not. We have not found any other molecule which has this kind of uniqueness. So what we thought that hydrophobic substrates are notorious for protein adsorption. So this is the schematic. This is the CH3 SAM, the ball model, and these are the CH3 groups on the top. This is alkane thiol SAMs. This is gold substrate. These are sulfur atoms. So we thought that the hydrophobic end of the protein molecule of this TMA molecule can be utilized for the molecule to self-assemble on the hydrophobic surface. Because the length scale of this molecule and the CH3 groups on the top is equivalent, so it can also bury itself into the interstitial spaces. So that was the idea, and that was the initial thought, that we can have all the O's pointing upwards, and we can make it hydrophilic, and since these hydrophobic interactions are small, we can actually remove it off. So to test this hypothesis, we used quartz crystal microbalance measurements, and just quickly, let's go over the technique. So it's a piezoelectric quartz crystal with a gold coating, and now it vibrates in shear mode, so it vibrates sideways at a resonant frequency. If molecules adsorb onto this crystal, the resonant frequency shifts, and the shifts in resonant frequency can be related to the mass adsorbed. And this is a very high precision balance. It can measure masses up to nanograms. It's similar to the technique surface plasmon resonance, but it measures the wet mass, where SPR measures the dry mass. SPR measures using refractive index of the protein layers, so it measures just the protein content. But here, whatever comes on the top, it measures that mass. This is how this mass versus time curve looks like. So this is the time of the sample injection. Adsorption occurs at a high rate because it just starts absorbing. and then it equilibrates over a period of time. So this is the